So last week we started talking about Mendelian genetics, and we never really got into how does that apply to us as humans. So we've already looked at the basics of our law of independent assortment and what genotypes and phenotypes are, and our F1 and F2 generation. And we've definitely looked at some crosses with peas, for instance, which is really how Mendel discovered all these things. But let's talk about how this applies to us. So human genetics follow the Mendelian model. And so here are just a few things, a few single traits that we can look at, whereas the dominant traits are freckles, the widow's peak that we've talked about, and a free earlobe. So do you have freckles or a widow's peak or possibly a free earlobe? The recessive traits are no freckles, a straight hairline, and attached earlobes. I actually have attached earlobes, so I guess that means my mom and dad both contributed recessive genes for me to have that. I do have freckles and I do have a widow's peak. Okay, so let's look at this just for freckles, for instance. So let's say that mom and dad are both heterozygous for freckles. So that means, let's say that mom up here contributed a dominant and a recessive allele. And then dad over here contributed, again, a dominant and a recessive. So what we would get out of that is a homozygous dominant, a heterozygous dominant, another heterozygous dominant, and a homozygous recessive. So that means we have a 1 to 2 to 1 genotypic ratio. But what is the phenotypic ratio? Remember, the phenotype is what it looks like. And for that, we have 1, 2, 3 that are dominant, so displaying freckles, and 1 that is not. So our phenotypic ratio is 3 to 1, just like we expected with the Mendelian genetics of the pea plants. Okay, so let's say then that mom has no freckles. So she is going to give us 2 egg cells. Oh, you know what I just realized? I did this backwards because this is a sperm, right? So, all right, let's say this is dad then. So sorry, dad. So dad, I guess, has no freckles, and we are adding 2 recessive sperm. And then mom then is going to add a dominant allele and a recessive allele. And when we look at the Punnett square for that, we have freckles, freckles, no freckles, and no freckles. So a 1 to 1 phenotypic ratio. So in other words, half of the offspring would have freckles and half would not. So the problem with human genetics is that you can't just do multiple crosses over and over and over again. First of all, our life generation is way too long. That would take years and years and years. And secondly, it's really not ethical to say, let's see how many we can produce out of you. So instead we do what's called a pedigree analysis. So that's looking at your family tree and tracing a particular trait. So let's say, for instance, we're going to use this as our key. The women are circles and the men are squares. And the circle is going to be a woman that does not have the trait, so an empty circle, no trait. And the filled in circle does have the trait. Same thing with the man. If the square is empty, he does not have the trait. If the square is full, he does have the trait. Okay, so let's look at the family pedigree of this lovely family. We're going to call them the Hartmans. And we are going to look at a trait that deals with the heart muscle. And in the dominant form, they produce a healthy heart muscle. In the recessive form, there's abnormal heart muscles that cause problems. So in order to have that heart defect, you have to have a homozygous recessive trait for this. So when we start, we know, hang on, sorry, I'm trying to get my pen. So we know that Joshua and Abigail right here have heterozygous alleles for this heart muscle gene. For John and Hepzibah, which is a lovely name, you know she's pretty, we know that they don't display the abnormality. So we know they have at least one dominant allele, and we don't know about the others. So Joshua and Abigail have 2 children. One of them has the heart defect. So we know that Jonathan has a homozygous recessive genetic. We don't know about the other child. We just know that she does not have the heart defect. So John marries somebody named Elizabeth, and Elizabeth is heterozygous for the trait. And how do we know that? We know that her parents did not have the defect, so she has at least one of the dominant genes, one allele. But when she and Jonathan have children, some of them show up with the defect. So that means she had to at least contribute one allele of the recessive trait. So that's how we can trace the family pedigree when we're looking at our family tree. And so here are all the children. So now that we know Jonathan and Elizabeth's alleles, then we can trace out the rest of their children and know which ones are heterozygous and homozygous. And we know that the ones that don't have the defect have to be heterozygous because Jonathan was homozygous, so all he can contribute is the homozygous, is a recessive allele. So that means that each of these children have at least one homozygous recessive allele. So that brings us to this point with Jonathan and Elizabeth's children. Since all of them have at least one recessive allele for the heart defect, they are what we call carriers. So that means that they have a normal phenotype because they still have one of the dominant alleles, but because of this recessive allele, they are a carrier. That means that they can pass it down to their children. So a carrier is a heterozygous person who carries a disease allele. So one of Jonathan and Elizabeth's children, named Charlie, is going to come in with being a carrier, and he is going to then marry somebody named Angie, and they are going to have children. And if Angie is also a carrier, then what we have then is our normal Punnett square with a 3-to-1 ratio, which means that 75% of their children are likely to have a normal heart, whereas 25% of their children, or a 25% chance that if they have a child, it will have a heart defect. Okay, so with Charlie and Angie's offspring, we said that there's a 75% chance that they will have a normal child and a 25% chance that they will have a child with a heart defect. But in addition to that, when we look at this chart, we also note that there's a 50% chance that their child will be normal but will be a carrier also. Cystic fibrosis is one of those examples of an autosomal recessive disease. So in cystic fibrosis, there is a particular channel protein for chlorine that is defective. And so those who have a homozygous recessive C allele then will have cystic fibrosis. So when we look at autosomal recessive pedigree charts, this is what it's going to look like. And you can tell this person right here would be affected by the disorder. Somebody like this is going to be a carrier. And again, here we go, 2 that are affected, and we know that it's an autosomal disease because it's male and female, so it doesn't matter who the sex of the carrier. And so that is how we look at that mapping of the autosomal recessive disease. Okay, so what about dominant autosomal diseases, where the dominant trait means that you're expressing the disease, whether you are homozygous or heterozygous, and the only way to escape that genetic disease is by being a homozygous recessive. Well, here are just a couple of examples of that. We have CHARGE syndrome, Marfan syndrome, hypercholesterolemia, sorry, I can't say that very well. And this one in particular is quite fascinating. Polydactyly. This is having an extra finger or toes, and it's actually fairly common in humans, dogs, and cats. So those of us who have 5 fingers and 5 toes actually have a homozygous recessive trait. Let that sink in for a minute. So here we go. If we are looking at our Sam and Susie, and they are both heterozygous, if they are heterozygous for the disease that is extra digits, that means that 75% chance that their children will have extra digits, and only 25% chance that they will be normal, in quotes, that only have 5 fingers and 5 toes. So far everything we've talked about has been pretty straightforward, genetically speaking. So let's just real quickly go through some variations on Mendelian genetics, because it is a little more complicated than just straight dominance and recessive, for some genes anyway. So one of the variations is what we call incomplete dominance. That means the phenotype is a mixture of the dominant and recessive phenotypes. If you'll remember, this goes back to the common thought of the day before Mendel even came up with his theory of the particulate inheritance. So this means that, for instance, our dark brown horse here, the dominant gene is the capital B, and so if we have 2 of those, we're going to have a dark brown horse. And then by nature, the recessive trait is a white horse, so lowercase b, lowercase b. But when we have a heterozygous, capital B, lowercase b, we wind up with a tan horse. So incomplete dominance, a mixture of the 2. And we see this phenotype in plants as well. So if you've ever seen pink carnations, that is because of the crossing between a red carnation and a white carnation gives us incomplete dominance, so we get a pink carnation. Interestingly enough, we can also have what we call codominance, which means both of the alleles are expressed at the same time. That's usually a bit rarer, but in that case we would have a red and a white being the dominant and recessive, and then when they're both expressed, we have a red and white petals both on the same flower. So that's codominance. Another example is the brindle pattern, in dogs. That is a codominance pattern. Now, another variation is what we call multiple allelism. So instead of having just your typical 2 alleles for each gene, there are multiple versions, and our red blood cells are great examples of that. So we have 3 different allele types, an A, a B, and an O. And so red blood cells are identified as A, B, and O based on the glycoproteins on the outside of the cell. And so I don't know if you know your blood type. My blood type is A, and so I am right here. And each person has 2 different alleles. But there are 3 alleles total as options for your blood type. So that is called multiple allelism. And then there's polygenic inheritance, and this is one that I find fascinating. And it's one trait that is the result of an additive effect of more than one gene. And so skin color is a great example of this. So how many genes are responsible for skin color? Many, many, many. And they all contribute to give a spectrum of skin color. I think it's fascinating. Here are some other traits that require multiple genes, so other polygenic traits. Not only our skin color, but our hair color, our height and weight, our eye color. So, for instance, I have very dark brown eyes, and I have one daughter who has super dark brown eyes. That's Claire. But my oldest daughter has very light brown eyes. And then I actually also have a daughter with very light blue eyes. Yes, my mother has blue eyes, and my husband's mother had blue eyes. But it is a polygenic trait, so it's more than just one gene. And so I like to think of it almost like dosage, like how many of those genes are turned on determines how much pigment you get in those in either your eyes or your hair or your skin. So I tend to call that dosage dependent, but it's technically called polygenic. Okay, so chromosomal inheritance. What we've been talking about so far has all been a part of our autosomal chromosomes. And most of our chromosomes are autosomal, right? 22 pairs of them. But there's one pair that are sex chromosomes. And so through meiosis, we wind up with half the DNA. So here we go with our 23 and our 23 and our 23, and these are all our sperm cells right here. And so when we look at our male germ cell, we have X and Y chromosomes, whereas our female germ cell is going to have just the X chromosomes. So when we divide those by meiosis, in our males, we'll have, percentage-wise, 2 female carrying, two sperm carrying X chromosomes and 2 sperm carrying Y chromosomes, whereas all the egg cells are going to carry just the X chromosomes. Okay, so the point is that the sperm determines the sex of the offspring, and you have a 50-50 chance of either having a boy or a girl. Now I'm just going to throw a caveat in there just to let you know that there was a study in the early 2000s that determined that the egg cells kind of had a say-so in which sperm cells they let in, chemically speaking, of course. So in that sense, yes, the sperm cell does determine whether you're going to wind up with a male or female offspring, but there is some selectivity based on the egg cells. But for now, let's just say we cross, like we normally do, our X and Y and our X and X. So in this, then, we would have the possibility of girls right here, 2 of those, and then 2 possible combinations for males. So this is where we get our 50-50 or 1-to-1 ratio of male versus female offspring. Okay, so then besides just determining the gender of the offspring, what types of genes are on these chromosomes? On the X chromosome, obviously is genes for being female, but also genes for everyday human function. And the Y chromosome actually carries very little genes, but just genes for being male. So the development of an embryo into a male. So because genes for everyday function that are on the X chromosome can carry diseases with them, the genes on the X chromosome are called X-linked or sometimes called sex-linked. So here are some examples of the genes on the X chromosome. One is a gene for blood clotting protein. When we carry the recessive, that results in a condition called hemophilia. So the inability to clot your blood. Secondly, there's a gene for a muscle protein called that results in a condition called Duchenne's muscular dystrophy. And again, that's a recessive condition. And thirdly, and probably the most fun really, is a gene for an eye pigment protein. And the recessive condition of this one causes colorblindness. So when people have muscular dystrophy, that means that in their muscle tissue, if they carry this abnormal gene, it means that they start to begin to have fibrous tissue within their muscles. And that makes them stiff and unable to be used as well. So they're not as flexible like you would expect your muscle tissues to be. So we actually have a good friend whose son has this, was in my oldest daughter's playgroup, and they didn't really notice until he hit about 2 years old, because that's when you start doing a lot of your muscle development and developing your motor skills, and that's when it becomes evident. So the way we denote genes that are sex-linked is if it is a dominant allele, we would put the X with a capital A. That indicates that it is sex-linked on the X chromosome. If it is a recessive allele, then we have the X with the lowercase a. So obviously for a woman, if they are dominant for any of these diseases that we talked about, because these particular diseases only appear when you have a homozygous recessive gene, then if it is a woman that carries a homozygous dominant for these, then we say no disease here. We can have a sex-linked heterozygous genotype, which means you have no disease but you're a carrier. And then the sex-linked homozygous recessive, of course, means that it's a woman with a disease. Now, if we're looking at a man, then we have to include the X and Y chromosome, and we only have the genotype written by the X chromosome because it's X-linked. And so a man with a dominant sex-linked gene would have no disease. A man with a recessive sex-linked gene would have the disease. So let's talk about colorblindness for a minute, because that's just a quick, easy, genetically inherited, quote, disease. And so if we're looking at a woman, a man, that's not what I meant to say, a man because he has a Y chromosome, but let's say this man is dominant, and that means that he has normal vision, and then we're crossing that man with a woman who is a carrier for the colorblindness gene. So when we do the Punnett square for their offspring, here we have a woman with no color blindness, another woman with no color blindness, a man with no color blindness, and a man with color blindness. So our percentages then mean that we have a 25% chance of having a child that is colorblind, right? Because only 1 out of 4 is colorblind. The percentage of having a boy that is colorblind is 50%. And the reason we say 50% is because we have 2 boys and 1 out of the 2 have color blindness. The percentage of having a girl that is colorblind is 0% because we have 2 girls, 0 of which have color blindness. So here is your red-green color blindness chart. So you should be able to look at this image, and if you have color blindness, you will not be able to see the number that is present. If you are not colorblind, you will be able to see the green number. And so I'd be interested to see which of you are colorblind and which of you are not. So please send me an email and in the subject say colorblind-yes or colorblind-no. And then we'll get together a chart from our particular class of how many of us are colorblind and how many of us are not.